Assalamu alaikum dear students. Today we are going to discuss all the chemical reactions that are involved in the citric acid cycle. The citric acid cycle is a series of chemical reactions that takes place in the mitochondrial matrix. This cycle uses acetyl coenzyme A derived from the sugar and fat breakdown to form ATP, NADH, FADH2 and carbon dioxide. The NADH and the FADH2 can be used to form additional ATP through the chemical reaction of the electron transport chain. The citric acid cycle goes by many names including the tricarboxylic acid or the TCA cycle or the Kreb cycle. The citric acid cycle or the citric acid refers to the citrate that is produced in the first step of the pathway. The tricarboxylic acid titles get its name from the 3 carbon dioxide that are produced from each fully oxidized pyruvate. The Kreb cycle refers to Hans Adolf Krebs, who identified the full cycle in 1937. He was eventually awarded the Nobel Prize for the Physiology of Medicine in 1953 for his discovery. In the first step of the cycle, an enzyme called citrate synthase joined the 2-carbon acetyl group from the acetyl coenzyme A with the 4-carbon oxaloacetate to form 6-carbon citrate. In the step 2, an enzyme called aconitase converts the citrate into isocitrate. Next, an isocitrate dehydrogenase enzyme oxidizes isocitrate, a 6-carbon molecule, to 5-carbon alpha-ketoglutarate. The carbon that was lost is released at carbon dioxide, and 1 NADH is also formed. The carbon dioxide that is released was originally part of the oxaloacetate and not acetyl coenzyme A. In the fourth step, an enzyme called alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate into 4-carbon succinyl coenzyme A. Similar to the step 3, in this reaction produce 1 carbon dioxide and 1 NADH. In the step 5, a succinyl coenzyme A synthetase enzyme converts succinyl coenzyme A into succinate. This produces GTP, which is converted to ATP in the 6th step, number 6. An enzyme called succinate dehydrogenase converts succinate into fumarate. This step makes 1 FADH2. In step 7, a fumarate hydratase enzyme converts fumarate into malate. In the final step of the citric acid cycle, a malate dehydrogenase converts malate back to oxaloacetate. Like all the steps involving in dehydrogenase, a coenzyme is produced. Here it is NADH. The oxaloacetate that was regenerated through the citric acid cycle is now all ready to join with another acetyl group and begin the cycle a second more time. For every 1 glucose that is broken down through glycolysis, 2 pyruvates will be produced and 2 pyruvate will produce 2 acetyl coenzyme A. So for every 1 glucose, 2 acetyl coenzyme will be made and 2 turns of the citric acid cycle will occur. This means that each product of the cycle must be doubled. A total of 4 CO2, 6 NADH, 2 FADH2 and 2 ATPs are made through the citric acid cycle. NADH and FADH2 are the electron carriers that can produce more ATP later in aerobic respiration. In addition to the sugar like glucose, protein, fat can also provide carbon substrate to fuel the citric acid cycle. The protein can be broken down into individual amino acids such as alanine, aspartate and glutamate and convert into intermediate in the cycle. Fatty acid can be broken down into acetyl coenzyme A, which then begin the citric acid cycle. This metabolism of sugar, protein and fat through the citric acid cycle provide the vital energy necessary to maintain many cellular respiration processes. I hope you like the video. Thank you very much for watching.